So our first speaker today is Dr. Cristen Willer. Dr. Willer is an associate professor with appointments in internal medicine, human genetics, and computational medicine and bioinformatics. Dr. Willer has become really a leader in using biobanks such as MGI to understand the genetic basis of cardiovascular disease and improve ways to treat it. So please welcome Dr. Willer.
So Sebastian asked me to talk about how me and my research team have been using MGI, and I was like, how haven't we been using MGI? So I'm going to try to cram into my 15-ish minutes 5 use case examples. So strap yourselves in, hold on, I'm going to go super fast. Just kidding, kind of. First, to make sure I have time to do this, I want to thank the team. So I'm including Lars in this picture because this picture was taken when Lars was sitting on our side. Now he's come over to the brighter side. But this is the work of all of these people. You know, the joke goes, I do my research in PowerPoint. These guys are the ones who are actually coding and hands-on with the data. So really, this is all of their stuff. So hopefully speaking to the choir, why do we study the genetics of human diseases and traits? I don't know if Gonzalo's seen this slide, but he'll probably recognize his brother as the example of a healthy young man. So the goal of our research is to encourage everyone to live healthy, active, long lives. And on the other side is my grandfather, who's 95 this year. With the idea that if we identify genes that are involved in a trait or a disease, these become leads for prevention and potentially treatment. And now we're moving into the area where we can hopefully predict individuals who might benefit the most from preventive lifestyle changes, medical screening, or specialized treatments, or just general treatments. So as you know, I was hoping all these things would come. There we go. Biobanks amortize the cost of GWAS across traits. MGI is a cohort collection of something like 45,000 genotyped individuals, samples in the bank on the order of nearly 70,000? 80? 70,000 samples, 80,000 consented people. There's limited amounts of frozen buffy coat. This is sort of a QC step of MGI, so if you catch something fairly quickly in the DNA, it's possible they might have frozen buffy coat in the freezer. But generally, MGI is a resource of collected DNA samples and a variety of phenotypes, primarily coming from ICD-based medical records and the entire EHR. And MGI has also, because they've genotyped 45,000 people with an array, has a collection of sort of quality control imputed genotypes available for researchers to use. So my first use case, my first example, is a situation where when I was hired into the medical school, my chief said, I really want you to collect a biobank focused on aortic aneurysm, aortic disease. And the first question we had to ask when designing that biobank was, do we want to spend the effort and resources collecting controls? And we made the decision to collect cases only because we're based in the CVC, we have lots of cases. That was the purpose of the study, with the idea that we would partner with MGI and use MGI samples as controls. So, and in particular, we wanted to study some phenotypes that can't be captured by ICD codes. So we use this as sort of a boutique biobank to supplement what's already in a large biobank that exists at the university. So one of the exemplar phenotypes that we wanted to study was bicuspid aortic valve, which is the most common congenital heart defect. It's a fairly mild congenital heart defect as far as congenital heart defects go. Something like, it's estimated up to 1% of the population walks around with this silent sort of minor change in their aortic valve where they have 3 leaflets that open and close instead of 2. And so we have probably one of the best and highest number of samples collected for bicuspid aortic valve globally because it's very, very difficult to diagnose in people unless they're having a valve replacement. So really the best place to ascertain these patients is in aortic surgery clinic where they're having a valve replaced, and that's where these samples were collected. So Because this is more of a workshop, I'm focusing a little bit less on the scientific discoveries and a little bit more on how the nuts and bolts of actually using MGI worked. So I'll race through the conclusions and move on to the issues that sort of arose and how we dealt with them. So just a quick overview of the study design. We have 466 cases of bicuspid aortic valve, which doesn't seem like a lot, but again, this hasn't really been studied in large numbers before. And we selected controls from the MGI biobank. And Wei Zhou, who was a PhD student at the time, is now a postdoc at the Broad, performed all the association analysis. So just to summarize the findings, cut to the chase, we performed a GWAS, had very well-controlled lambdas in the end, 2 genome-wide significant findings, which were the first 2 to be found for bicuspid aortic valve using this genome-wide GWAS study. The most interesting, most strongest hit was a non-coding variant near GATA4, which is a key cardiac transcription factor, which if you had to pick a candidate gene ahead of the study, GATA4 sort of sits there with flashing neon lights around it. We also noticed, and I remember the moment I looked at this plot with the graduate student, that there's a coding variant with a significance of around 10 to the -4 in GATA4. And upon following both of those variants up, the non-coding and the coding, they both replicated in subsequent studies. We performed some, we looked at some Hi-C data to demonstrate that the non-coding variant does loop and interact with an intron in GATA4, likely impacting transcription of that gene. And we completed some in vitro studies. This is through work of a collaborator, Bo Yang, to demonstrate that disruption of GATA4 disrupts the ability of mesenchymal cells to differentiate into, endothelial cells to differentiate into mesenchymal cells, which is a key step of cardiac valve development. So the story is good, published in Nature, Santi was here, Nature Communications, I think. Yeah. And what made this study work well, aside from not having to collect controls, which would have halved our sample size because we would have spent half of our money collecting controls. What was key was that we genotyped our case samples in parallel to MGI. We used the same exact array in the same version, actually matters. We used the same laboratory, so the DNA sequencing core. We asked the sequencing core to, sorry, I don't know if you guys knew this, but we asked them to interject our plates intermittently within MGI plates to try and reduce batch effects. Not samples or anything. Just throw the plates in, do them on the same day. So they used the same reagents, and then we used the same imputation panel, the same quality control for our samples, because honestly, when we first did the analysis, without doing the same quality control, we found very inflated QQ plots. We found lots of false positives, and honestly, we spent 6 to 9 months trying to eliminate any batch effects between our cases and MGI controls. And what ended up making the biggest difference in the end was through conversations with the MGI team, we discovered that MGI had created their own cluster files. If anyone's done any clustering of genotype array data, the data comes out in sort of on an XY scatter plot, and it usually falls into 3 little clusters. And you can use pre-computed clusters from Illumina, which is what we were using, or you can draw your own based on your own data. And MGI had drawn their own. We were using the pre-computed, and any difference in the cluster files would result in false positives because our genotypes are being called differently. So when we used the MGI cluster files, thank you for sharing, on our own data, a lot of the false positives went away. We also found, because we were dealing with a small case number, 466 cases, but MGI had, at this time, 20,000 controls. We had very unbalanced design, and although we used principal components for matching ancestry, with such an unbalanced number of cases and controls, principal components might not be strong enough to account for any population differences. So we instead went in and within the PC space, calculated the distance between all pairs of samples in our study, the CHIP study, which stands for Cardiovascular Health Improvement Project, and MGI. And for each case in our sample, we selected the best matched control Then we did it again for the second best match control, and we ended up going as far as 10 ancestry matched controls to each case. So we had our 466 cases, 4,660 controls. I'm never going to get through 5 use cases. We'll see how far I get. 10-to-1 controls to cases, and this, again, really helped the lambda come down, and our association statistics looked well controlled. Good. Use case number 2. We actually went in and pulled from the freezer MGI DNA samples. So in this case, we wanted to perform exome sequencing of a small set of samples that had aortic dissection, with the hypothesis that there were novel or perhaps unique variants within these individuals causing, with high penetrance, aortic dissection. And so we wanted to, of course, use controls, and we selected controls from MGI. So again, this is just an example of what we have within our Cardiovascular Health Improvement Project Biobank, something like 5,000 DNA samples, around 750 aortic tissues. In this case, we were studying the subset that had previously had an aortic dissection. We selected age, sex, and ancestry matched controls. So using the GWAS array data, we could select a control that was closely ancestry matched. We sent them all for exome sequencing, and when the data came back, we took every variant identified within the 11 genes that were known to cause aortic dissection, and we sent all these variants to Diana Milowitz, who is the world's expert on dissection, aortic dissection genetics, and she annotated all the variants we saw in either cases or controls as pathogenic or not. And what came back was 25 unique pathogenic variants in 26 individuals. When we deconvoluted the cases from controls, we found that all 26 individuals who carried a pathogenic variant were in the dissection cases, and 0 were in the controls, which made us feel much more confident about that pathogenic variant annotation. We actually went on to evaluate, and we have a paper written about this, evaluate the implications for clinical care. So for 8 individuals, there were implications for their clinical care, either new diagnoses or altered diagnoses. And we actually opted to return these 26 non-CLIA genetic results to the participants. And we have just completed doing that last week. We used a very carefully thought out protocol where a genetic counselor on the research side called the participant back, returned the result, explained that it was not CLIA, could not be used for clinical testing. It's not in their medical record at this time. The patient controls whether that data will get into their medical record. And at that point, they can choose to have a CLIA test to validate the result, and then it becomes part of standard clinical care. What made this exome sequencing study work? Again, we used existing MGI and CHIP genotypes to select ancestry matches for the controls. We pulled MGI and CHIP DNA, and they were sent for exome sequencing at the same time, in the same lab, same reagents, et cetera. All DNA processing and variant annotation was performed blinded to case control status, which again made us more confident about the calls that we observed. Use case number 3. We used a new technology in-house through a collaboration with Jacob Kitzman to perform targeted sequencing. So in this case, rather than looking at all genes in a small number of people, we wanted to look at a smaller number of genes in many, many people. So we were piloting and trying to scale up a technology where we selected about 120 genes for targeted sequencing, and this technology is called MIPS, molecular inversion probe sequencing. It's basically like exome sequencing, but just for smaller numbers of genes, with the idea that it hopefully costs somewhere between $20 and $30 a sample. So to pilot this study, we selected equal numbers of CHIP cases and MGI controls. And again, pathogenic variant calls were made by an external sort of gold standard, as best you can do, calling variants as pathogenic or not, by Partners Healthcare in Boston. Again, Partners Healthcare doesn't know who's a case and who's a control. It turned out We're just starting to process the data, but included in our panel were the 56 ACMG genes, which was the ACMG list at the time we designed the panel. And what has just recently been returned to us is this table that of the around 11,000 individuals we sequenced, about 58 in CHIP have pathogenic calls for one of the ACMG genes, and 34 individuals in MGI. So at some point I'm going to very soon return these all to the MGI crew and let you guys figure out what you want to do about it. In particular, I saw Nick walk in, an anesthesiologist. He's quite interested in the RYR1 mutations. These are mutations known to cause malignant hyperthermia of anesthesia, which he knows about and I don't, but basically it's a bad thing that happens if you give them the wrong anesthetic. Do you want to say more?
Perfect.
Very bad thing? It's a very bad thing. So we now know of 3 people who are patients at U of M who we could just put a flag in their EHR and Nick and his colleagues could give them the right anesthetic. Wouldn't we want to do that? So that's use case number 3. What made this work? Same as before, careful matching of cases and controls, processing the DNA in identical manner, and blinded pathogenic carrier status. You know what I mean. Moving on, use case number 4, using MGI for methods development. One of the key questions we're looking at in our lab and many labs right now is, can we use genetic data to prevent disease, to identify individuals at high risk for disease and target specific treatments? So I'm not going to get into the nitty-gritty of polygenic risk scores, but basically they sum across many, many variants in the genome, thousands, could be millions of variants in the genome, each with a very small effect, and identify individuals who, through sort of bad luck of collection of those millions of variants, end up at the higher risk end of the distribution for a specific disease. It does seem to work in real life. For example, an optimized risk score for coronary artery disease, the individuals in the top 1% of the polygenic risk score distribution have an odds ratio of 5 times higher risk of getting CAD than the rest of the individuals. So if we can select those individuals, say out of the MGI data set, we could make sure they see a preventive cardiologist, maybe get on statins, and try and actually prevent disease. However, it's not that simple in real life. To use a polygenic risk score, the optimizing and testing really needs 3 independent data sets. You need one data set to generate your GWAS and all your weights for those millions of variants across the genome. You need to optimize the weights. Should you use 1 million variants? Should you use 1,000? Should you use 10? It's not completely obvious at this time. And then lastly, once you've decided on your weights and your optimized model, you need to test the prediction in a 3rd independent data set. And that's where we've been using MGI in our lab. So for example, your GWAS weights might come from UK Biobank. We optimize the weights in study sample number 2, another study we work with, and then you can test them in MGI study number 3. Last one, time for the last, gonna cut me off, is a standard GWAS. I won't go into this for much detail. Hopefully you'll hear more examples of this, but in our case we looked at atrial fibrillation, meta-analysis with many cohorts, summary of the findings, published in Nature Genetics, it's all great. What made this work well was collaborating with the MGI research team, so we understood very well where the imputation came from, what analysis methods were used, handling ancestry appropriately, etc. So in summary, I presented very quickly 5 use cases of how we've used MGI, both the genetic data and the DNA samples themselves, primarily as controls, but also for discovery for the many phenotypes that are available within MGI. Things to watch out for, just be very careful, as we all are careful scientists, about false positives. So to minimize this, we use the same array, including version as MGI. We used the same cluster files. We excluded any variants that differed between control groups in the 2 data sets. And we used a common quality control and imputation pipeline. One of the things we noticed in working with MGI samples for sequencing is that the DNA concentration may need rechecking. If you're doing a study with 10 samples, you can extract them by hand and do every DNA quantification under the sun. If you're working with 80,000, 70,000, it's much harder. So just to keep in mind, if you're going to spend a lot of money on an experiment, you might spend the $8 to $10 ahead of time requantifying the DNA. It's important to consider ethnicities. You'll hear more, but MGI is essentially 95% European ancestry. I think something that's really important when you're thinking in the rare disease space is to call pathogenic variants blinded to case control status, and I think using MGI as controls really made that study possible. And I think we should consider what to do with pathogenic results, whether we want to be talking about returning these to participants or not. Thanks.
Let's have a quick minute for a question.
Not really a question. I just want to echo your comment that we should think about what to do with returning results. So I believe that Dr. Pasternak will talk about a project a little bit later where we're running into the same thing on the pharmacogenetic side. We're having a very difficult time getting this through the IRB and figuring out, is this clinical care? Is this a new clinical trial that we're enrolling them on? So to hear that you guys are doing some of that, I'd be interested in hearing how you went through that process.
So briefly, it took us about 3 or 4 years to get a process in place by talking to legal, ethics, genetic counseling experts, medical genetics experts. Basically by hiring the cardiology genetic counselor on our research team, we are paying for her genetic counseling appointments. When she's disclosing the research level results, she is part of the research team. Everything at that point is on the research side. She then basically gives pre-test counseling to the participant, explains what this might mean for their clinical care and their family members, and then when she orders the CLIA test, as of right now, the research study is paying for it. I think that's too high a burden on a research study. I'd like to see that moved back into, that's a clinical test at that point. But then when that CLIA test comes back, the genetic counselor is now on the clinical side. That now goes into their medical history. Now their provider knows. Before that, I am not disclosing a research level result to any of the providers. So in that way, I felt more comfortable that the participant decides whether it goes into their medical record from a research study or not. That's the way we're opting to do it. Very happy to share protocols, scripts, anything that would help with that process. We're also studying the impact of the return on the participant. How stressed are they? Decisional regret, that kind of thing.
All right, thank you.